Chapter 42, Hemostasis and Coagulation Instrumentation. So we'll go over the different type of laboratory coagulation of instruments, advantages, disadvantages, all the methods that we're going to do to detect it, platelet function stuff, point of care, genetic testing. So the coag lab, always changing. Coag is huge because it tells us about thrombosis, tells us about platelet function, tells us a lot of things. So we have to have a lot of information to give the physicians to help treat. So I don't know how you pronounce that thing, but that's the first blood clot assessment instrument that was out there. Then we have TEG analyzers, which we'll talk about here in a minute. Nephelometric, electromechanical, and then how we've gotten better. So most of the assays we do are endpoint, and you've done them. You inject everything, put everything together, and it stops. Clot, boom. That's called endpoint. So they're totally automated, and then they're semi-automated. So they had different ways it's done, methods: mechanical, photo-optical, chromogenic, nephelometric, immunological. So there's limitations to each one of those, but there's been a lot of improvement since then. This is an real old instrument that we used to use called a fibrometer. The next instrument, the diagnostic histago, which is what we use in class. But they both are considered a mechanical endpoint detection. In other words, it measures the conductivity between, in the fibrometer, the metal poles, and then rods that go in. And then when we looked at you'll see in a minute on the next slide, the clot detection using a little bead going back and forth. So here with the fibrometer, it's got like one little wire, and it click, 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 and then it clots and the click stops. That's because we've got a clot made. So that was like the first big time way it could be done actually testing. So this is the method that we use, the clot detection in our lab. That's one of the ways, mechanical, is that, remember, it's an electrical magnetic field, and you inject your plasma, and you inject your reagents, and the steel ball goes rocking back and forth. So as the viscosity increases because of the clot being formed, the movement slows, eventually stops. Timer indicates clot time. This method is liked because we don't run into the methods when we talk about optical here in a second, the light going through. So lipemia, icteric, turbid stuff doesn't cause a problem. So you can look at the video, and we'll do that in class, but you can watch that video if you would like. But they're showing you the bottom right, the picture of it. Where the bead's moving back and forth, and you can see the little wave back and forth where the ball's moving back and forth, and eventually will stop. Another assay endpoint is optical. We look at the detection there. It's called turbidimetric is another name for it. So here we got a light. You inject the stuff into it, so a light's going through there. So it reads the transmittance of the light through, and then you add the stuff. And as it makes a clot, then the light going through changes, and the photodetector will pick it up. So it's looking at what they call the optical density, the OD. So it records it. So it looks at the clarity of it. So when a fibrin strands are formed, the clarity is different. The light gets scattered, so therefore it stops. So that's the Sysmex, the coag analyzer Sysmex makes. They use photo-optical. Another method is called the viscoelastic clot detection method, and this is actually looking for how the clot is made, and it actually grafts it. So you can see the grafting at the different angles. It's like a little wire that's in there, and it measures from mechanical tension how it's making it. I'll show pictures later on that. Another type of method is looking for color. That's called chromogenic endpoint. So they actually have, like here showing you fibrinogen, and then we add thrombin, and you can see there's a whole molecule there. So the enzyme thrombin will break it apart. That's called cleaving it. So then the substrate will then attach, and then we get a color change. Like protein C, factor 10, all use chromogenic. So here is really a spectrophotometer checking for the color, the chromophore being present through it. The other method is the immunological method. So we're looking at light absorbance because we're doing antigen antibodies. And it looks for cloudiness, nephelometric they call it. You talked about that clin chem. In other words, the light comes, goes through, and then when we got stuff in it happening, clots happening in this case, the light will scatter to the side, and we look at that. So you can see the pictures of that. The different sources. It can be straight through or off to the side. Side scatter, they call it. So this is, I know, very small, but it's in your book. So it lists the advantages and disadvantages of each. The biggest disadvantage we see a lot of times is the photo-optical, because it has interference with lipemia. That's usually one of the big problems that we run into with that. It might not detect a very short clotting time. You know, the mechanical really doesn't show the graph of the clot formation. So there's advantages and disadvantages in each. So you got to see, you know, what method you're going to end up using in your lab. So coagulation instruments have definitely gotten better. They got better accuracy and precision. The more automated you get, the better it's going to be, so you don't have to pipette. I mean, when you do semi-automated or manual, everything has to be done in duplicate. But when we get to automated, it's just single test, just done once. So you save half of your reagents right there. So one of the things we look at is called random access. In other words, each patient, you don't like do all the PTs and all the PTTs. You just, you know, each patient, you can do all the testing as you need it. Also, the instruments have improved reagent handling. In other words, stuff needs to be cooled where it needs to be cooled, refrigerated. Stuff needs to be heated where it needs to be heated. We track the reagents, make sure if we're running low, running high. It can do primary tube sampling, in other words, right out of the tube, or we can do closed tube where it can actually puncture, cap piercing, and go through it that way. It can also do automatic dilutions for you. So, I mean, that's the advancements that we're running into that make it a little bit better. And then they have nice computers that hold all that information for you. It can read the barcodes. It can look at the graphing that comes on. It can flag stuff for you. It can tell you what's wrong. It can tell you if the reagents aren't working. You know, so it makes it a lot easier, the more automated, because, you know, there's so much we have to look at now. So these advancements have really helped us with that. Now, this is the one I was talking about with the TEG, the thromboelastograph, so you need to really see that video. They have these in most of the hospitals around us now, and it actually sees how well the clot forms. It actually graphs it, so you can see it, so that's pretty cool. Another one is called rotational thromboelastometry, ROTEM. So whole blood is placed in the cuvette. They activate it, and then they put a pin in, and they sort of like rotate it around to see how the clot forms. You know, how the pin is moving in the cup. Seems like a fancy way, doesn't it? So platelet aggregometers. Again, this is huge for platelet function testing. If we don't have that, looking at it on a smear doesn't tell us much. Counts really don't tell us much. But we really got to look at the function. So that's why we have platelet aggregometers. So in this particular one, they're using platelet-rich plasma. And what they'll do is they get the platelet-rich plasma, and then they'll inject things that will make the platelets release or secrete the stuff in the granules, which will allow them to actually start like clumping. So you can see here some of the agonists or the stuff that's coming out of the granules, ADP, collagen, calcium, whatever the agonist may be. And you can see the picture, the platelets are suspended, and they're got a little stir bar in the platelet-rich plasma. And then you squirt in the agonist, and the platelets will then aggregate onto the electrode. And you can see it in the picture on the right. So that's how it measures that. And this some of the different names that we'll actually see with a lot of those instruments. So in most of the hospitals, we have this thing called a PFA 100. It's a platelet function analyzer. There's actually cartridges that will contain the agonists, either collagen, like we said, ADP, epi, that will stimulate platelet aggregation. So you get whole blood, and you put it in the aperture, and you measure the time it requires to make a platelet plug. So what it's doing is how long does it take to block the aperture? That will tell us a platelet function. So that's one. Verify now is another one. They usually lose little tiny beads around the platelets to look at them clumping, called agglutination. And then they have another one called platelet works. So it's actually looking on the platelet count before and after they become activated. That's another one. Another method that can be done is flow cytometry. So here, you actually get whole blood, and it will look at the platelet surfaces, how they become activated, how they bind to leukocytes, how they secrete, and how they aggregate. Because that's important in heparin-induced thrombocytopenia. So we got to see the functionality of the platelets, because usually when somebody has heparin-induced thrombocytopenia, the platelet counts really start tailing off. Not much you can do but get them off the heparin, but that brings another nightmare to the process here too. So then we'd have molecular testing, and we'll talk about when we get into the thrombosis chapters. But thrombophilia, in other words, you're clotting too much, so that's when you got to look like for antithrombins, protein CNS, factor V latent mutation. Well, like I said, we'll talk about that. Where there's a prothrombin mutation, the G2210A, it's not responding to antithrombin. So there's all kind of methods, to it. But again, to get to this stage, you've done a lot of testing to get there anyway. So with the molecular genetic testing, it gives us real fast results, like the Cepheid Gene Expert. It can do a lot of those testing. So molecular is definitely the way things are going, for sure. So look at those different methodologies. Make sure you know the difference between a mechanical and photo-optical. Okay? I don't care about selection criteria, but, in other words, what you look at for you buying a new instrument. So look at the summary, look at the questions, and look at the videos. And if you have any questions, let me know. Thanks.